Good morning, everyone, and welcome. I'm delighted to have you here today. Today, we'll be exploring the topic of microbiology testing, focusing on current expectations and requirements. This is an important area that impacts many aspects of our work and compliance standards. Over the course of this presentation, we'll cover key expectations, regulatory requirements, and best practices in microbiology testing. So, let's get started and delve into this essential subject. Let's start by understanding the current regulatory expectations for microbiological testing. It's essential to be aware of what regulators are looking for to ensure compliance and quality. We'll also discuss the key compendial chapters and global guidelines that set the standards across different regions. Moving forward, I'll outline the GMP requirements specifically for microbiology laboratories, covering the essential practices to maintain integrity and reliability. We'll review routine microbiological tests, focusing on their purpose and the acceptance criteria that define whether a batch passes or fails. It's also important to highlight the significance of validation processes, ensuring our methods are accurate and reproducible. Additionally, maintaining data integrity is crucial. Trustworthy data underpins regulatory compliance and quality assurance. Finally, I'll touch on risk-based approaches, which help prioritize efforts and resources to areas with the highest impact on product safety and quality. All of these elements together form the foundation of microbiology testing today, aligning with regulatory expectations and supporting overall compliance. Let's take a closer look at the global regulatory agencies that oversee pharmaceutical and biotech products. Key players include the US FDA, the European Medicines Agency, or EMA, the UK's MHRA, India's CDSCO, and the World Health Organization, or WHO. These agencies set the standards and regulations that ensure safety, efficacy, and quality across different markets. Moving on to GMP guidelines, which are essential for manufacturing practices. For instance, the EU GMP guidelines include Annex 1, which focuses on sterile products, and Annex 2, covering biologics, with detailed Parts I and II. In the U.S., the 21 CFR Parts 210, 211, and 600 provide the regulatory framework for pharmaceuticals and biological products. There are also harmonized guidelines that promote consistency across markets. The ICH, or International Council for Harmonization, has developed several important guidelines. Q7 addresses API GMP, Q8 focuses on pharmaceutical development, Q9 deals with quality risk management, and Q10 outlines the pharmaceutical quality system. These guidelines help streamline regulatory processes globally. Pharmacopoeias play a critical role as well. The USB, NF, the European Pharmacopoeia, JP from Japan, and the Indian Pharmacopoeia all provide standardized tests, including microbiological testing chapters, which are harmonized to support quality and safety. Overall, understanding these agencies, guidelines, and pharmacopoeias is fundamental to ensuring that our products meet international quality standards and can be safely used worldwide. Let's start by discussing the bacterial endotoxin test, or BET, which is covered under USB 85 and PH. EUR 2614. This test is crucial for ensuring that our non-sterile products are free from harmful endotoxins, which can cause serious reactions if present. It's a key part of microbial limits testing in non-sterile products, helping us assess safety and quality. Next, we have environmental monitoring, guided by USB 1116 and EU GMP Annex 1. This aspect involves monitoring the manufacturing environment to control microbial contamination. It's essential for maintaining a clean production area and ensuring our products meet the highest safety standards. Remember, depending on the market, we might need to adjust between USB, PH, EUR, or IP guidelines, so understanding regional requirements is important. Moving on to bioburden or microbial enumeration, covered in USB 61 and PH, EUR 2612. These tests help us quantify the total microbial load in our products, giving us an overall picture of microbial contamination levels. It's an important step to ensure that our products stay within acceptable limits. Then we have tests for specified microorganisms found in USP 62 and pH. UR 2613. These are targeted tests to identify specific microbes that could be particularly harmful, such as pathogens. Detecting these precisely helps us manage risks effectively. Finally, sterility testing, which is outlined in USP 71 and pH. UR 261. This test confirms whether our products are free from viable microorganisms, especially for sterile products. It's a fundamental part of microbiological quality control to ensure patient safety. All of these chapters and tests form the backbone of microbiological quality assurance in pharmaceutical manufacturing. They help us maintain compliance, ensure product safety, and ultimately protect patients. Connecting these points to our overall theme, they highlight the importance of a comprehensive microbiological control strategy across all stages of production. Ensures microbial safety of drug products. This is a critical aspect because it directly impacts patient health and trust. It provides supporting data for key stages like product release, where we verify that each batch meets safety standards. It also underpins stability studies, helping us understand how products maintain their integrity over time. Additionally, microbiology plays a vital role in process validation and cleaning validation, ensuring that manufacturing processes consistently produce safe products. This role is directly linked to patient safety and regulatory compliance, making microbiological control an essential part of our quality system. Furthermore, microbiology acts as a feedback loop for facility and process control, helping us identify and address potential issues quickly. Overall, the role of microbiology in pharmaceutical quality is fundamental to delivering safe, effective medicines and maintaining our commitment to high standards. Let's take a closer look at product testing, which is a crucial part of ensuring quality and safety. First, we have bioburden and microbial enumeration tests. These are used to determine the level of microbial contamination on a product, helping us assess its cleanliness before it reaches the patient. Next, we perform tests for specific pathogens. These are targeted tests to detect particular microorganisms that could pose health risks. For sterile products, sterility testing is essential. It confirms that the final product is free from any viable microorganisms, ensuring safety and compliance. Bacterial endotoxin testing is also important, especially for parenteral products and some medical devices. This test checks for harmful endotoxins produced by bacteria, which can cause severe reactions if present. Moving on to utilities and support systems, these include purified water, water for injection, clean steam, and compressed gases. These utilities are vital for manufacturing processes and maintaining product integrity. Environmental monitoring, or EM, involves regularly checking air quality, surface cleanliness, personnel, and pressure differentials. This helps us maintain a controlled environment, reducing contamination risks. Finally, validation and qualification encompass verifying that our methods are suitable, disinfectants are effective, and media used in testing are validated. These steps ensure that our processes and tools consistently produce reliable and accurate results. In summary, microbiological testing isn't just about compliance. It's about safeguarding patient health by ensuring our products are safe, sterile, and free from harmful microorganisms. Let's take a closer look at the microbial limits outlined by USP 1111 and pH. UR 514. These standards specify the acceptable levels for microbial contamination in non-sterile products. First, we have the total aerobic microbial count, or TAMC, which refers to the total number of aerobic bacteria present in the product. This limit is specific to each product, depending on its intended use. Next is the total combined yeast and mold count, or TYMC, which measures the levels of yeast and mold. Again, these limits are tailored to each product, ensuring safety and quality. Importantly, the standards also require the absence of certain microorganisms that can pose serious health risks. For example, Escherichia coli, Staphylococcus aureus, Pseudomonas aeruginosa, and Salmonella species are all considered critical microorganisms that must not be present. For some products, Candida albicans is also specifically tested for. The limits for these microbial counts and the absence of specific microorganisms are defined based on how the product is used. For instance, whether it's administered orally, topically, through inhalation, or parenterally. The route of administration influences the microbial limits because it affects how the product interacts with the body. Additionally, the patient population is a key consideration. Products intended for neonates or immunocompromised individuals, for example, require stricter microbial limits due to their increased vulnerability. So, in summary, these microbial limits are carefully established to ensure product safety, tailored to the route of administration and the patient group, aligning with overall quality standards for non-sterile pharmaceutical products. When it comes to sterility testing, we follow specific methods outlined in USP 71 and PH. UR 261. These include membrane filtration and direct inoculation, which are the two primary approaches used to assess sterility. Key requirements for these tests are quite strict. For instance, testing must be conducted in a Class A or ISO 5 environment, ensuring the highest level of cleanliness. This typically means working within a qualified cleanroom or using laminar airflow cabinets or isolators to maintain aseptic conditions. Additionally, the processes involved must be validated, including sterilization and depyrogenation, to confirm they are effective and reliable. It's also essential to include both negative and positive controls during testing to verify the accuracy and integrity of the procedures. I want to emphasize that sterility testing is really a verification tool. It's not meant to replace a well-designed sterile manufacturing process or robust aseptic controls. Instead, it acts as an additional safeguard, helping us confirm that our processes are working as intended and that the final product is sterile. Overall, these regulatory expectations are in place to ensure the highest quality and safety in sterile pharmaceutical manufacturing, aligning with our broader goal of protecting patient health. Let's take a closer look at the methods outlined in USB 85 and PH. UR 2614 for bacterial endotoxin testing. These include several approaches such as the gel clot, turbidimetric, chromogenic, and recombinant factor C methods. Each of these techniques has its own advantages and specific applications, but all aim to accurately detect endotoxins in pharmaceutical products. Now, regarding regulatory expectations, it's crucial to understand that endotoxin limits are defined based on the dose and route of administration. This means that the acceptable level of endotoxins varies depending on how the product is used, which ensures patient safety. A key point is that any method used must be validated. This means it should be reliable, reproducible, and capable of detecting endotoxins accurately. Validation involves additional testing for inhibition or enhancement to confirm that the test results are not skewed by other components in the sample. Using traceable endotoxin standards is also essential. These standards serve as a baseline to ensure consistency and accuracy across different testing batches and laboratories. Finally, trending bet results over time is a best practice. It helps us monitor the ongoing process performance, identify trends, and ensure continued compliance with quality standards. All these elements together form a comprehensive approach to bacterial endotoxin testing, which is vital for ensuring the safety and quality of our pharmaceutical products. Now, let's take a closer look at the regulatory drivers shaping our environmental monitoring programs. Key among these are the latest revisions of the EU GMP Annex 1 and USP 1116. These guidelines set the standards for how we should monitor and control environmental conditions in pharmaceutical manufacturing. When we talk about environmental monitoring components, or M components, we're referring to several critical elements. First, we have non-viable particles, which we count online to monitor airborne particulate matter continuously. Then, there are viable particles, which we assess through active air sampling and settle plates to detect living organisms in the environment. Surface monitoring is another key aspect, using contact plates and swabs to evaluate cleanliness on surfaces. Personnel monitoring, such as finger dabs or garments, helps us understand contamination risks introduced by staff. Data collection is fundamental. We use this data to identify trends, set alert and action levels, and support batch release decisions. If deviations occur, the data helps us investigate root causes effectively. A significant shift happening now is moving towards risk-based, scientifically justified environmental monitoring programs. This approach allows us to focus resources where they are most needed, enhancing both compliance and product quality. Overall, the current focus in environmental monitoring is on integrating these regulatory requirements and scientific principles to create robust, proactive programs that safeguard product integrity and patient safety. Let's start with facility and design. It's crucial to have controlled access to the microbiology lab, ensuring only authorized personnel can enter. We implement unidirectional flow, moving from clean to dirty areas to prevent cross-contamination. Segregation is also key. Dedicated spaces for sterility testing, BET, and routine work help maintain the integrity of each process. Moving on to equipment, we rely on qualified incubators, biosafety cabinets, laminar airflow hoods, autoclaves, refrigerators, and particle counters. All of these are regularly monitored and maintained to ensure optimal performance, which directly impacts the quality of our work. Regarding reagents and media, vendor qualification is essential, and we verify certificates of analysis to confirm quality. Additionally, we perform growth promotion testing on all media to ensure they support microbial growth as expected, which is vital for accurate results. In terms of documentation, maintaining thorough records is non-negotiable. We keep standard operating procedures, test methods, data sheets, logbooks, and calibration and maintenance records. These documents support traceability and compliance with GMP requirements. Finally, our microbiology laboratory must adhere to GMP standards, ensuring consistent quality and safety in all testing activities. Every aspect, from infrastructure to documentation, is designed to uphold the highest standards in microbiological testing. Let's talk about the regulatory expectations we need to meet. First, it's essential to demonstrate that our methods are fit for purpose using the actual product. This means showing that our testing methods are suitable and reliable when applied directly to the product we're working with. When it comes to microbial enumeration and specific microorganisms, we need to perform a method suitability or recovery study. This is done in the presence of the product to ensure our methods can accurately recover and detect the microbes despite any complexities the product might introduce. It's also critical to demonstrate that there is no significant antimicrobial interference. In other words, our product should not inhibit microbial growth or interfere with the detection process, which could lead to inaccurate results. Regarding validation parameters, we focus on accuracy, which is essentially recovery, how well our method can detect the microbes when they are present. Precision is about consistency, ensuring that repeated tests give similar results. The limit of detection tells us the smallest amount of microorganism our method can reliably identify. And robustness checks that the method performs well under small variations in conditions. For BET, or biological effectiveness testing, we need to do an inhibition or enhancement test with the product matrix itself. This ensures that the product does not artificially suppress or boost microbial counts, which could mislead our assessments. Overall, this slide emphasizes that both method validation and method suitability are key. They form the backbone of ensuring our microbiological testing is accurate, reliable, and compliant with regulatory expectations, ultimately supporting the quality and safety of the product. When it comes to sample collection, it's crucial to use validated sampling procedures for all the key areas: product, water, environmental monitoring, and utilities. This ensures that the samples truly represent what we're testing, giving us reliable and accurate data. We also need to make sure our sampling is representative in every way by selecting the right locations, choosing appropriate sampling frequencies, and collecting the correct volumes. This helps us get a complete picture of the conditions we're monitoring. Aseptic handling is another critical component. It requires trained personnel who follow strict aseptic techniques and use disinfected materials to prevent contamination. Proper use of personal protective equipment, or PPE, is essential too, to maintain the integrity of the samples and protect everyone involved. Controlling hold times and transport conditions is just as important. We need to minimize delays and ensure samples are kept under appropriate conditions to avoid false results, which could lead us to incorrect conclusions about quality or safety. In summary, thorough sample handling combined with aseptic technique is fundamental to obtaining valid results. Everything from proper sampling methods to handling and transport plays a role in supporting our overall quality assurance efforts. Let's talk about the regulatory focus on ALCOA plus principles, which are fundamental for maintaining data integrity in microbiology. These principles emphasize that data should be attributable, meaning we can trace who recorded the data and when. It should be legible, so it's clear and readable at all times. Contemporaneous recording is crucial, documenting results at the time they are generated to prevent any memory issues or alterations later on. The data also needs to be original, meaning it's the authentic record from the source, and accurate plus complete, ensuring the data reflects the true results without omissions or errors. Consistency and enduring storage are key. Data should be stored securely and remain unchanged over time. Finally, the data must be available when needed, easily accessible for review or audits. Now, the key expectations linked to these principles include avoiding backdating or rewriting results, which can compromise data integrity. Any corrections or deviations should be controlled, properly documented and justified. Electronic systems like LIMS must maintain audit trails, so every change or access is recorded, ensuring transparency. Raw data, including plates, printouts, logbooks, and electronic files, need secure storage to prevent tampering or loss. Regular data integrity audits are essential, along with ongoing training for microbiology staff, to reinforce these standards and ensure everyone understands their roles. Ultimately, maintaining data integrity in microbiology isn't just about compliance. It's about ensuring trust in our results and supporting the overall quality of our work. Let's take a closer look at the risk-based approach to microbiology testing. This approach is grounded in principles from ICH-Q9, which focuses on quality risk management, and Annex 1, which guides sterile manufacturing practices. The first key point is identifying microbiological risks across all critical areas, including raw materials, equipment, utilities, the environment, personnel, and the process itself. It's important to have a comprehensive view, so we don't miss any potential sources of contamination. To effectively evaluate these risks, we use tools like FMEA, which helps us analyze failure modes and their effects, HACCP, which is excellent for pinpointing hazards and establishing control measures, and risk ranking and filtering methods to prioritize the most significant risks. These tools allow us to make informed decisions rather than relying on assumptions. Once we identify and assess the risks, we can use that information to design targeted sampling plans, setting appropriate alert and action levels. This means we'll know when a result indicates a potential problem and what steps to take next. It also helps us determine the right frequency and extent of monitoring, balancing thoroughness with efficiency. Finally, the risk assessment guides us in prioritizing CAPA, corrective and preventive actions, focusing our resources on the issues that truly matter most. Overall, adopting this risk-based approach ensures that our microbiological testing is strategic, proactive, and aligned with our overall quality management goals. when we encounter out-of-trend or out-of-specification results in microbiology, such as elevated counts, the presence of specific organisms, or a failed sterility or BET test, it's crucial to understand that these are indicators that something may be off in our process. The first step is to conduct a thorough investigation, starting with phase I, which involves checking for lab or analytical errors. This helps us rule out simple mistakes or technical issues. If no errors are found, we move to phase II, where we perform a full investigation that looks into the process, utilities, environment, and even personnel. The goal here is to identify any underlying factors that could have contributed to the out-of-specification result. A key part of this process is root cause analysis, where we might use methods like the 5 why or fishbone diagrams to dig deep into potential causes. Once we identify the root cause, implementing corrective and preventive actions, or CAPA, is essential. We also need to verify that these actions are effective in preventing recurrence. Throughout this process, documentation plays a critical role. It must be thorough and aligned with regulatory expectations, such as those outlined in FDA OOS guidance. Proper documentation ensures transparency and supports compliance, especially when regulatory agencies review our investigations and results. In essence, handling OOS or OOT results involves a systematic approach, careful investigation, root cause analysis, CAPA implementation, and meticulous documentation. This structured process helps us maintain the integrity of our microbiological testing and ensure product quality and patient safety. Now, let's talk about some of the increased emphasis we're seeing in the industry. First, isolator technology and closed systems are becoming more critical. These systems help minimize contamination risk by creating a more controlled environment, which is especially important for sterile manufacturing. Next, we have RMM, or rapid microbiological methods. These are innovative testing approaches that can deliver faster results than traditional methods, but they do require thorough validation and acceptance by regulatory bodies before they can be widely adopted. We're also seeing a push towards real-time environmental monitoring and data trending. This allows us to get immediate insights into the environment, identify trends, and act proactively to prevent contamination events. Contamination Control Strategy, or CCS, remains a cornerstone for sterile products. It's about implementing comprehensive, layered controls to keep products sterile throughout production. And all these elements, CCS, quality risk management, product quality systems, environmental monitoring, utilities, and microbiology testing, are increasingly expected to be integrated holistically. This means they should be interconnected, with each component supporting and justifying the others, providing a more robust and transparent approach to quality and compliance. Finally, in terms of regulatory trends, authorities are now more focused on these integrated, real-time, and validated approaches. They're encouraging us to adopt innovative strategies while maintaining rigorous validation and documentation. This overall shift is shaping how we approach microbiology and contamination control in our industry. Let's take a closer look at some of the common regulatory observations we encounter in microbiology testing. First, inadequate or unvalidated environmental monitoring programs are a significant concern. If these programs aren't properly designed or validated, they can fail to detect potential contamination risks, which compromises product safety. Next, poor documentation and incomplete data integrity controls are often seen. This can lead to gaps in traceability and accountability, making it difficult to verify that testing was conducted properly. Ensuring robust documentation is critical to maintaining compliance and confidence in the data. Inadequate GPT or media control is another issue. This refers to the management of growth promotion tests and media used in microbiological testing. If these controls are weak, it can lead to unreliable results that don't accurately reflect the microbiological status. Equipment that's unqualified or poorly maintained also poses a big risk. Proper qualification and regular maintenance are essential to ensure equipment performs consistently and results are reliable. Then, we have inadequate investigations into microbiological failures. When a contamination or failure occurs, thorough investigations are vital to identify root causes and prevent recurrence. Skimping on this step can jeopardize product safety and regulatory compliance. Lastly, relying on non-scientific justifications for alert or action limits is problematic. Limits should be scientifically justified and supported by data. Arbitrary or unsupported limits undermine the credibility of the testing program. All these points highlight the importance of a well-designed, validated, and scientifically sound microbiology testing program. Addressing these common issues helps ensure product quality, safety, and compliance with regulatory expectations. Let's talk about some of the best practices for ensuring microbiological compliance. First, the strong training and qualification of our microbiology staff is absolutely essential. We need to make sure everyone is well-trained and continuously qualified to stay up to date with the latest techniques. That includes periodic requalification, especially for critical practices like aseptic techniques and gowning, to maintain high standards of cleanliness and contamination control. We also rely on robust SOPs, standard operating procedures, that are regularly reviewed and harmonized with the latest compendial updates. This helps us stay aligned with regulatory expectations and industry best practices. Another key point is frequent trend analysis of microbiological data. By continuously monitoring trends, we can identify potential issues early before they become serious problems. Regular internal audits and mock inspections are also vital. They keep us prepared and ensure our processes are compliant at all times. And finally, we adopt a proactive approach using quality risk management, QRM-based improvements. This allows us to identify risks and implement preventive measures proactively rather than just reacting to issues after they occur. All of these practices together reinforce our commitment to compliance and help us maintain the highest microbiological quality standards in our operations. Microbiology testing plays a vital role in ensuring the quality and safety of pharmaceutical products. It's a highly regulated area, meaning we have strict oversight to make sure everything is done correctly. Compliance in microbiology is driven by several key factors. First, we follow current pharmacopoeial chapters, which set the official standards for testing and quality. Second, we adhere to global GMP guidelines and ICH-Q series, which provide a consistent framework for quality management across the industry worldwide. Third, data integrity and risk-based approaches are essential. They help us ensure the information we generate is trustworthy and that we're proactively managing potential risks. Having strong systems in place is crucial, along with well-trained people and validated methods. These elements are the foundation that helps us ensure patient safety and maintain regulatory compliance. To summarize, effective microbiology testing isn't just about following rules. It's about building a comprehensive, reliable system that supports high-quality, safe pharmaceutical products. For further support, please contact us. We're here to help and ensure you have all the resources you need. Thank you so much for your time and attention throughout this presentation. If you have any questions or need additional assistance, don't hesitate to reach out. We appreciate your engagement and look forward to supporting you moving forward.